All right, so we've got our muscle contraction halfway finished. If yours isn't out, you're not listening to me. I need you to listen. Turn this off for a second. All right, so can we see that on the video? Yeah, okay. All right, so yesterday we started this. Let's review a little bit. We're going to start the nervous system today. That's why I gave you those notes. All right, so what is this? Blue guy, the fist.
Postsynaptic.
Not postsynaptic. It'd be presynaptic. What part of this other neuron are we looking at? Is this a dendrite? Is this an axon? Is this a cell body?
Terminal.
The axon terminal, the end of the axon. Okay, if you need to write that in there, do it. I'll go ahead and write it on this one.
This all?
Yes.
What do you want us to do with these?
Oh, I need to pick them up. Just leave them in the middle right now. All right, so this is the axon terminal. Okay, what type of neuron is this if it is attached to a muscle? A motor neuron. Okay, so this is a motor neuron. The motor neuron is going to carry out what part of our neural pathway? Remember, our neural pathway starts with what? Input. What type of input?
Sensory.
Sensory input. Okay, that would be being blind. I'm trying to feel my way. Okay, in case you were wondering what that was. So, sensory input, then what happens? Integration. Who does integration? The brain. Then what happens? Motor output. So that's what this motor neuron is responsible for. Causing the desired reaction that the brain determined was appropriate in the body. Okay? Alright, so we have just depolarized our last node of brain VA. It should be followed by the influx of calcium to repolarize it, but instead we get what? We get calcium gates opening instead, because we're at the last node of brain VA and we want to push these bubbles, which are called what? Synaptic vesicles. We want to push these bubbles all the way to the edge so that they can attach with what types of proteins? Snare proteins. What's going to happen to these snare proteins? They're going to rip open like Hulk Hogan, okay? And by the process of what, these neurotransmitters are going to get across the gap? By diffusion, okay? We've created a concentration gradient, lots of neurotransmitters here, not any over here. So they're going to diffuse across the gap, which is called a synapse, and they are going to enter what portion of our sarcomere? The T-tubules. Remember, this is like the sewer system around our muscle fibers. Okay, so these T-tubules are running everywhere. And if you watched Big Guns, and if you didn't watch it yesterday and you still haven't watched it, I should get to hurt you. Okay, if you watched Big Guns, it showed you a really good picture. The T-tubules were yellow, I believe. Okay, they were yellow, and they were just little tubes that ran down all the muscle fibers. Okay, so this neurotransmitter, by concentration gradient and diffusion, is going to diffuse across the synapse and enter the T-tubule. The signal that that neurotransmitter sends is going to trigger what? Not depolarization. We're not in a nerve anymore. We're in a muscle. What'd you say, Katie? Calcium. Calcium is going to come from where? Sarcoplasmic reticulum. That is the equivalent of what organelle in general? The ER, the endoplasmic reticulum. What's the endoplasmic reticulum's job? To move stuff. It's the highway. It's the chemical reaction site in our body. So this sarcoplasmic reticulum is then going to diffuse calcium out, and calcium is going to go flooding into these thick and thin fibers. Okay? Now we're going to zoom in on this. Okay? Will someone take a picture of this and tweet it to me?
I got you.
Okay, will you take a good one, like get up and really get it?
I selfied it.
Can you even see the picture?
No, but it's a really good picture of you, though.
Am I really annoyed?
No, yeah, I was going to take it to them.
Thank you, Becca. All right, just tweet it to me and I'll tweet it out.
That's all you want to see Paula Dean ride something?
No.
Tweet it to
That
me.
sounded really bad.
You can
Well, I
tweet
mean,
it to me.
there's like 900 of these.
If you go to my Twitter feed, is that how you say it?
Yeah.
Okay. I just followed a site called Injury Pics.
Oh, I follow that.
It's pretty intense. Pretty intense. Yesterday they posted a guy who was a hockey player that got a skate to the face, and he had a big gaping, like, all the way across the face. 52 stitches. So, it was pretty cool. They show a lot of, like, hey, look what happened to softball today, and this finger's pointing the wrong way and stuff. It's pretty neat. All right, so, you don't need to follow them, but when I see really cool ones that aren't that gross, I'll send them to you.
Yeah. No gross. I just tweeted at you.
Thank you. See, now I know that you're supposed to say at you and not to you. Didn't know that
That's
the
why
other day,
there's
and my
an
ninth
at
graders
sign.
were like, it's at, not to.
Well, it's an at sign.
Because they know so much.
Yes, the ninth graders are so cool.
I was a ninth grader last year.
All right. Woo! It's sad, my arm gets tired from erasing the board.
You got it, yeah.
All right, so we're going to draw a big picture here. All right. The skinny fibers. So these skinny guys here, and I'm going to actually draw them kind of big. But compared to the other ones, they won't be that bad.
Is this what we're drawing at the top?
Yep, you're drawing this across the top.
Can I get another paper? Yes. She said you can. Is that at the top of it? That's what I want.
I'm going to draw just one set of thick and thin.
Miss Hole, where's your paper?
It's in my printer.
Wow, that was a really delayed reaction.
That's my camera. Scratching the arm a bit. Yep.
I'm just gonna wait. So how did you hear me all the way back here?
I'm a teacher.
Senses. She has a baby.
Okay.
Baby equals superpower.
So, here's our picture. This would be the end of one sarcomere there. Okay? So there'd be another sarcomere here and another one after that. And these would be alternating. I'm only going to draw one set. But you would have another orange and another green and another orange and another green. Okay? And they're separated like this. The oranges split, that's not perfect center, but the oranges split center and the green is lined up right there. And they're not really green and orange, but... Alright, are we ready?
Yeah.
Get ready.
Can we draw this like really big?
This is how big mine is. This is mine.
Oh my god.
Okay, and if you watch Big Guns, this should all seem familiar.
That
Okay.
thing was confusing.
Okay, so these are like our kids at the dance. The boys are represented by the myosin, so you can put its name on here. Because myosin wants really, really bad to touch the actin. Hey, jibber jabber, zip it. All right. So they want, myosin really wants to touch actin. Actin's just fine hanging out. Right, ladies? Okay. So the ladies up here and the boys down here want to touch each other. But we got to build up the courage.
That's
Okay?
really not how the middle school dances work.
I'm sure. They're all over each other.
They keep like.
Now, we've got to identify... We got to identify some portions of this actin before we can get moving. These little guys up here are called myosin heads. Think of these like fingers, okay? They want to reach out and touch you, all right? All right, so on the actin, we have a couple little things we got to look at. We have these ropes, and it's kind of hard to draw 2D, but this rope is going to wrap around the actin all the way across. Okay?
Candy cane.
Kind of, yeah. This rope is called troponin.
I was thinking Halloween.
And it'll be on this side. Okay. Now on troponin, kind of, oh sorry, that's the wrong one. Tropomyosin.
Are you serious?
Oh, darn it!
Like a perfectionist, go ahead and crumple up your paper.
I'm sorry. All right, now on tropomyosin is troponin, and troponin is holding this tropomyosin in place. So this one is troponin, I'm sorry. this is representative of young ladies' better judgment. Okay? Now, underneath, where you can't see right now, but underneath this rope, what this rope is laying on are these little myosin binding sites. Now, we can't get to these binding sites right now because the lady's better judgment is holding tropomyosin in the way. Okay, but if we can get that better judgment out of the way, then we'll open up the binding sites and there will be some, hopefully just nice touching. Okay, maybe some dancing, we'll say that.
Wait, so
They're not working yet.
That's what you would call it.
Kind of, yeah, kind of like that. The better judgment, the chaperone, whatever you want to call it.
In my middle school dances, they told us to keep Jesus Christ between us.
So these little bursts are the myosin binding sites. And myosin wants us to expose those sites. Troponin is like, heck no. 6-foot bubble, right? All right. So this is inside, and now if we had like a layer that we could lay on top of this, if I had some big saran wrap, we could draw our sarcoplasmic reticulum and our T-tubule on top of this. Okay, they're kind of on the outside. This is deep inside the muscle fiber. All right, but I don't have any special saran wrap to do that, so just imagine.
It'd be awesome.
It would be really cool. All right, on the computer, when he shows the video, he actually can do that because they have like animators, and I don't have that. Hence the expert markers.
That'd be kind of cool if you like when we do like the bilayers and stuff.
Yeah.
If you could like do that.
That would be really cool. Maybe I can come up with something next year. Maybe someone sells sheet plastic or something. Oh, I might be able to use like drop cloths that are anyway. Let's keep going. All right. So what has happened so far on the outside of this? We had the action potential come in. We signaled the neurotransmitter vesicles to open. They diffused across the gap. They went through the T-tubule, and then what happened? The calcium, okay? So calcium is being flooded into our myofibril here. Well, calcium is the distractor. Need another color, red. All right, so what calcium is going to do is it's going to jump on board with troponin. This is our little calcium 2+. Okay, so calcium's going to jump on board with troponin and be the distractor for this chaperone and this better judgment. If you're thinking better judgment, this might be, well, not middle school dance, but this might be alcohol. Okay, if you're in college and 21. Okay. If you're still thinking middle school dance, this might be a fight that breaks out. Okay, all the teachers go look at that, and the kids go make out on the other side of the gym.
Or somebody
Okay.
spikes the punch.
Yeah, someone spikes the. Does that happen in middle school?
Yeah, those punch spike in old school.
That's
It's
crazy.
bad.
All right. So what calcium is going to do?
Go to a good school.
Calcium's going to bind with troponin, okay, and it's going to cause an action. The action is it's going to cause the rolling of this tropomyosin rope out of the way. Okay, and I don't really know how to animate that, so you can just draw an arrow. So when calcium binds, all of these ropes are going to slide just a little bit off of these binding sites and expose those binding sites. Okay? Kind of cool, right?
That's what it all goes down.
And that's when it all goes down. It gets a little crazy after that. Okay? So draw the arrow in the same color as you drew calcium so you know that it doesn't happen until calcium gets there.
So the ropes on.
The rope's going to kind of roll out of the way and the binding sites will be exposed. Okay? At the same time, or not exact same time, but when that happens, these myosin heads, or these grabby little fingers, are going to recruit some ATP. And ATP is going to be a star. And that's really not showing up as much as I thought it
I
would.
thought you were writing like a permanent marker.
Cannot even see it. Those stars.
Those are stars. Let's do it in, well, one green's already up there. Let's do it in orange.
That's sick. It doesn't say.
Okay, so ATP gets recruited because we can't really do anything in our body without ATP. So ATP comes in and builds up the courage of these little middle school boys. And what happens is, please don't be gross about this, they're going to do what's called the cocking mechanism. And they are going to perk up, okay, and reach for these binding sites. Okay, we shouldn't have used the middle school reference. Okay. All right, so what's going to happen is these little, they're called myosin heads or myosin cross bridges. They're going to tilt. So instead of being just relaxed and laid back, they're going to tilt up like this, and they're going to reach for those binding sites. So this one will reach for that one, this one will reach for that one. Now, once we use up that ATP, remember ATP is used up by doing what to the molecule?
Taking a phosphate.
Taking a phosphate. So once we use up that phosphate, we've got to recruit a second ATP to get them to relax. So when they reach like that, this whole thing gets shortened. Okay, so they're going to pull on those sites when they do their cocking mechanism. Okay, so when they go from here, you can see how this would shorten. They're going to grab on and shift. Does that make sense? Let me see if that's on camera. Am I on camera?
Now you're on camera.
Okay, they're going to, this is the little myosin head, okay? It's going to grab onto that binding site and then shift. You can see how my arm is flexing, right?
Huh.
Giant
You
guns.
can't see it.
Okay? All right? You can see it, look right there. All right? Just a small change, all right, in definition. So that cocking mechanism is going to pull these actin fibers towards the center of our sarcomere. Does a muscle lengthen when it's contracting? It always shortens. So that is exactly what's happening here. So in this sarcomere, when that cocking mechanism occurs, it will shorten this sarcomere. Same thing here. This one's shortening. They're all shortening at the same time together. Does that make sense? And they're getting more compact. That's why a flexed muscle, is it squishy? It's hard. Because you're packing all of these fibers into one place, into a smaller area. Yes?
So what happens when you pull a muscle?
You've torn, like, way larger scale than this. You've torn multiple fibers.
So all that happens to like this.
Just to go like this. Just to go like this. Just to do this. Okay? Any muscle in your body. Somebody yesterday in 9th grade thought the brain was a muscle. I said, what's the most important muscle in your body? He said, the brain!
That's why they don't know anything.
Yes. Okay. So, are we following here? What's happening? All right. In your space that's left over, I want you to outline the steps. Okay?
The
Your heart.
Yeah.
Okay. So, let's start number one. Number one, and you're going to build these together right now. So, number one, impulse travels through the final neuron to the what? What do we call this? In general, muscle or gland? Well, I mean, what are muscles and glands? Effector cells, okay? So the impulse travels through the final neuron. That final neuron is the motor neuron to our effector cell. Because this could also be a gland. The gland is not going to contract, per se. It's going to cause another reaction. But, yes, impulse travels through final neuron. If you want to put motor in parentheses, you can. But it won't always be a motor neuron because if it's such a gland, that's not going to be motor. To the effector cell. In this case, our effector cell is a muscle. All right, now we're going all the way back to the other picture. Once we get to the effector cell, what's the first thing that has to happen? Calcium's not first. What triggers calcium? Okay, what's in the T-tubule?
Little
Neurotransmitter. So stop number 2. Neurotransmitter diffuses through synapse and does what? Travels down the T-tubule. That's fine with me. Through the synapse. I might leave out a couple non-necessary, non-important words.
Starts its journey
Down the T-tubule. And if you want to put sewer system or whatever, if you want to add your little bits to it, you can. All right, so the T-tubules are full of neurotransmitters. What's next?
Calcium goes through the
Okay, so calcium is triggered to be released from the sarcoplasmic reticulum. Okay, write it all the way out.
How many steps are there?
I have 6 on here. Some people put 7. There might be 5.
35.
What's step
Okay.
3? What is that?
Step 3. What was step 3? We just
Calcium.
put our neurotransmitter through our
Leak.
T-tubule.
Through the things.
Okay, calcium's going to leak through what things? Sarcoplasmic reticulum.
How do you
Write it out at least once. It was on the board. Just like it sounds, pretty much. Sarcoplasmic and reticulum you've seen before. All right. So we've released calcium through the sarcoplasmic reticulum. What's next? We're on this picture now.
Then the
What's calcium going to do?
better line.
Good. Calcium, you can say distract if you want to stick with our analogy, or you can say binds with. Who's it going to bind with? Troponin. And you can put this in the same step, or you can move to another step. What's troponin going to do?
Move the ropes.
What are the ropes? Okay, so troponin, being distracted or bonded with calcium, is going to slide the tropomyosin, or the rope, and I put rope on there so I wouldn't forget, out of the way. When it slides out of the way, what is it now exposing?
The binding site.
Good, it's going to expose the binding site. So that may be 2 steps, that might be 1 step, that might be 3 steps for you. You decide how to break that down. So calcium did what? Calcium binds with troponin. What does troponin do?
Tropomyosin.
Moves tropomyosin, exposing the binding sites. We're almost done. Okay, now that the binding sites are open, what's going to happen? Myosin heads do what? Say it. She's like,
They perk up.
They perk up, okay? We call it the cocking mechanism. Okay, think of it like, I'm going to show you my muscle. It's just like that.
Wait, what?
The myosin heads. Now, what do they use in order to do the cocking mechanism?
ATP.
ATP. Make sure that's on there. Okay? Remember, locomotion, second largest user of ATP in the body. Okay? Make sure you include the use of ATP. Now, the part we didn't really go through, and you can add this kind of as a star at the bottom, if this movement to contract required ATP, do you think they're going to be able to lay down just by themselves? No. What are we going to need again? ATP. Okay? So once they're attached and contracted, we can't let go unless we have ATP. What does that tell you about life? Do you use energy to relax your muscles? Yes. So if you don't have enough energy, what's going to happen to some of your muscles?
We're gonna get stuck.
They get stuck. What do we call that?
A cramp.
A cramp.
Those things hurt so bad.
Okay? So it's not just dehydration and loss of ions and electrolytes. It's also, did you eat enough? Do you have enough ATP in your body to let that muscle relax? What happens when you die? Rigor mortis. He talked about this in the video. If you're dead, do you have enough ATP to relax your muscles? No. That's why you get rigor mortis. Okay? Your muscles that had previously been contracted can't relax, and so your muscles stay firm. Okay? That's why if you're, this is morbid, but if you're murdered and buried in a trash can like this, it's going to be real hard to fit you in a casket. Okay? They'll be able to do it, but they're going to have to like flatten you out. It's going to be tough.
Put a book on you.
Yeah, they might be able to press you like flowers. Okay? All right. So, we good on this? After 3 days of trying to get to this?
Yes.
Okay. Last step, once we've done the cocking mechanism with the myosin heads, you can add, or just put a star at the end, it can be your last step, however you want to do it. Another ATP molecule is attached to cause relaxation of the myosin heads. So each little myosin head, and think about how many myosin heads you're going to have in one sarcomere. This is like 7, 8 layers thick. Each little head needs its own ATP. That's a lot of ATP to just cause one muscle to contract. Right now I've got multiple muscles contracting to hold me upright. Okay, not to mention the smooth muscle that's digested my peanut butter and jelly sandwich for this morning. It's the only thing I can eat that sticks with me till third. It's the only thing.
Did you throw up a lot of
No, I still haven't been sick, but when I eat
just
something,
waiting,
it's like it's just gone. It's like fluff. All right, we good?
Why are you pregnant do you throw?
The hormones in your body just set you off. They do crazy things to you. And your stomach is just super sensitive. All right. Here's your homework that's due on Monday. Okay, I'm going to show you a picture of it. Write this down somewhere. Let me find it. I know I've got a picture of it. Here it is. Nope. Quinn's cheat sheet number 1. You guys remember Quinn, right? Oh, I didn't take a picture of the front. Let me show you the front. You are going to create a foldable. Woo! Right? Yeah. Really exciting. Okay, so here's the parts that need to be on your foldable. This is what it's going to look like. You're going to take a piece of paper and turn it hamburger. Okay? And you're going to create 2 doors. And you're going to cut 3 flaps on this door and 3 flaps on this door. On the front, you need to hit the 6 main pieces from this unit. I'm going to list them now. Okay? Hearing and balance.
Is that in one?
That's all one. Yep. Hearing and balance. Action potentials. Muscle contraction. So we've got hearing and balance, action potentials, muscle contraction, then neural pathway. That's your sensory integration motor. Last 2 are locomotion and synapse. Now on the inside, and let me hit the light. I'll say them one more time. Hearing and balance, action potential, muscle contraction, nervous pathway, or neural pathway, excuse me, synapse, and locomotion. So for example, what is this one on? Vestibular, cochlear, tympanic. That's hearing and balance, okay? So you can draw a picture over here. She drew a little flow chart for herself so she could remember. She gave all of this information that she could. Anything that she thought was important, she put on the inside. For muscles, she drew the close-up version of the 2, the thick and the thin fibers. Okay, second largest user of ATP. She gave me a flow chart of what's going to happen step by step. On, that's as far as down it's going to go. On locomotion, she just didn't draw a human, I guess. That's a pretty good chart. She's better than me. Okay, she talked about each one individually. Let me go back to the other one. Her bird was pretty cute. Here's her synapse. We drew it a little different last year. Okay, so she just took the whole area to draw the whole thing out. There's her action potential. I really like this number the different pieces. Okay, and then she went through and did 1, 2, 3, 4, and 5 over here. All right, and then the last one was the neural pathway. She drew her neuron, labeled everything, and talked about the different pieces. Okay, so this is what I want to see on Monday. Queen Kong, yes. Okay, I want to see this Monday. And I will not post these pictures. Okay, won't do it. I will post a... I think I have a PowerPoint slide of like, this is what I need you to do for homework. So I will post that PowerPoint slide. I'll take a picture of it and post it on Twitter, and I'll put it on my blog. Okay, can you handle that? You realize how to do this? Wide sheet, make some doors. Okay, do Monday. Monday, because I want to know that you studied. And this is the biggest, easiest way to study. Yes, ma'am.
I don't know if this is like a stupid question.
Ask it anyway.
Do you not know
What about like the parts of the brain?
No, that was just for the hat. Yep. We're going to start immune system right now. Okay? All right, pull out your notes for today. I know I only have 5 minutes, but we can get started at least. We only have technically 3 pages of notes left, and the last one's really short. So we're going to kind of fold them all into one. This would be day 8. Day 6 was one of those essays that they put in the middle. Day 7 is the lab we're going to do when you guys get back from Christmas with the crickets. Oh, I moved the camera. When did I do that? It's been filming the desk. I think I just moved it. Okay. Yeah, I did. Now it's on my hand.
Yes.
This is day 8, but that's really like day 6. Okay. Because we skipped 2 days in there. All right, so the immune system. Remember things from other units at the top? You got glycolipids and glycoproteins. These are all proteins in your membrane that are going to identify your cells as self. They are also going to be receptors for certain ions and certain gated channels. You know that the sodium-potassium pump is only for sodium and potassium. Every little thing in your body has its own little pump. Also, you need to remember that a ligand is a what? Signal molecule. Okay? All right, so your innate immunity. Your innate immunity, what's that word innate mean? If I say, this is an innate behavior. Nothing, huh? Nobody knows? You're born with it, okay? You didn't have to be taught it. The opposite of innate is learned. Okay, it was a learned behavior. Your dad walks around touching his stuff all the time. You see the little boy walking around touching his stuff all the time. Okay?
So, like, blinking?
Blinking is an innate behavior.
Breathing?
No one had to tell you that. Okay? An innate behavior is being afraid of snakes. Okay? For some reason, even little babies who don't know what a snake is are afraid of snakes. Okay?
Is that new?
Growling dogs. They don't know that a growl means that that dog is mad, but they can figure it out really fast. Okay?
Just like instinct.
Instinct is a good way to put it. Okay? But when we're talking about your innate immunity, we're talking about that immunity that you're born with. Now, if everyone is born with the same immunity, you think it's going to be very specific?
No.
No. It's going to be kind of general blanket statements. All right? So things like your skin. Everyone's born with skin. Skin is the first line of defense. All right? What's in your nose, in your mouth, and in your genitalia? Mucous membranes. Okay? Those mucous membranes are there to catch any foreign invader if they can. Okay? What do you think boogers are?
Foreign
All right.
invaders.
Foreign invaders. Dirt, dust, bacteria. Okay? So if you know anyone picks their nose and eats it, tell them not to.
That's
Okay?
disgusting.
Really gross. All right? That's all that is. It's the mucus that lines your nose being combined with dirt and crud. Okay? Crud. That's scientific. Crud. This also includes your sweat. Okay? Your sweat is mostly salt, but it does have a chemical called urea in it. What is urea?
It's in cat pee and cigarettes.
It's in your pee.
It's in everybody's pee.
Okay? So technically, you're semi-peeing out your body.
That's weird.
Okay? When you're sweating.
Yeah, but sweat.
It has some of the same chemicals. That's why sometimes when people get really sweaty and then they walk by you, they stink. Okay? That kind of BO smell, that's from urea. Right? It's also from not washing, but it's not washing the urea off. All right, this also includes the normal bacteria that you're born with. We call it the flora in your gut. Remember we talked about babies are born with certain amounts of bacteria in their gut. Where do they get it?
Oh my goodness.
Some of them are developed in the womb, but very few.
In the canal.
The canal, remember when you're covered with lactobacillus bacteria when you come out of your mommy's vagina. Okay? If you were a C-section, where did you get it?
You got your back up.
No, wait, we talked about this the other day.
If you were a C-section,
Like a glaze.
where could you have gotten some of your bacteria?
From the food.
Don't make me shimmy.
Breast.
Breastfeeding, okay? I'm really glad I didn't have to do that to cue you in.
I was thinking.
All right.
Don't make me shimmy.
All right. This also includes saliva, tears. Think when you get something in your eye automatically waters. That's part of your innate immunity. You will have white blood cells. Okay, white blood cells are called phagocytes. What is phagocytosis?
It's the eating in a cell.
Okay, the eating of a cell. So we call them phagocytes because that's how white blood cells work. They have these little cytoplasmic extensions, almost like pseudopodia from amoebas, and they can reach out and grab a cell that's bad and pull it in, and they can engulf it. Okay, they just swallow it whole. Anything? I'm gonna try and get 1, 2, 3, and 4. All right. So these are white blood cells that eat pathogens. Most of them are going to be neutrophils. Okay, just regular old neutral. Okay, they're just going to be general. Some of them will be bigger, what we call monocytes. Okay, these are larger macrophages. We call them macro. What does macro mean? Big. Okay, they're still phages that are going to swallow things, but they're bigger things they're going to eat. Let me talk about chemotaxis before the bell rings. That's number 3. Okay, chemotaxis is the ability to move toward a chemical or away from a chemical. Positive chemotaxis is you sense a chemical and you want to move towards it. Okay, what do we know in our body, especially men, that senses a chemical and swims towards it? Sperm, right? They sniff it out with their nose. They have chemoreceptors in the tip of their little spermy heads, and they swim towards the egg. All right, and then the last one is who breaks down all the bad things in our body. Once the macrophage goes, what's inside the macrophage? Lysosomes, okay? So lysosomes and peroxisomes are going to have those digestive enzymes that are going to break them down. So chemotaxis is a big one that we're going to see in the immune system. You're going to have positive and negative. Okay, so knowing the difference. Chemo means chemical, taxis means movement. So movement based on a chemical. Positive is towards, negative is away. Okay, that's a huge one. All right, and then the ability to know that a macrophage is going to swallow something is good too. They technically swallow it, then spit its head out and put its head on a stake. It's kind of Braveheart-ish.
Is that your favorite movie?
Yes. All right, have a good day. We'll finish this and start the next one tomorrow. Thanks for hitting the lamp.
You're welcome.
We're almost done with this unit. Almost done. Don't forget about your homework, please.
You said that's due Monday?
Monday, yes. Not due this week. But you do have a quiz tomorrow, don't forget.
Oh, you said it was chi-squared.
No, gives free energy. Chi-squared is Friday. Have you taken chemistry?
What is it?
Yeah, you're good. Yeah, this is kind of giving you a bird's eye view. Yeah, I think you'll like it. And if you take chemistry, you're perfectly set up to take it. Right, some people will take chemistry and this class together. Those are like the Emma, what's in this one? Emma Fell, put it up now. She decided against it because she kind of laid it out and was like, no, it'll work better in the thinking later. I wouldn't let her do that. But as a junior, senior, if you take chemistry, you're ready to go. Okay, well, I mean, you've got 2 years to decide if you want to take it. And if you like chemistry better, take Chem AP. Okay, and then when you take physics, you might want physics AP. So, I mean, there's other sciences to choose from, but, I mean...
This is my
Okay, well, good. I'm glad I could be your favorite.
Bye.
Bye.